Hello and welcome to our presentation on the ecological restoration of the bull at Cove State Park. My name is Valente Ayala. I'm a student in the Masters in Conservation and Restoration Sciences program here at UCI, and I'm one of the 4 team members who worked on this project. During this presentation, I'll be going over the background and methodology for this project. Maryam will go over our statistical analysis and results for the coastal sage scrub experiment. Peter will discuss our results on the coastal sage scrub experiment, and Justin will wrap it up by presenting on our herbivory experiment. However, before we go over any of that, we wanted to acknowledge that this campus and Cove State Park is built on the ancestral lands of the Yahachman and Tonga peoples. We would also like to thank our partners, Cove State Park, Cove Conservancy, and UCI CEB. Cove State Parks has been heavily impacted by humans throughout history to today. These impacts include cattle ranching and farming in the past, and current impacts on Cove State Parks include invasive plant species establishment and changes to the rain and drought cycles. The importance of this study can't be overstated. Monocultures of invasive plant species, typically black mustard, provide a low-quality habitat for native animal species. Restoring the site will provide our native animals a wider breadth of plant species to live in and to feed on. Additionally, this project will also provide key information to Cove State Parks on how to best implement management plans for this site. Our native plant communities have also been shown to perform better than invasives during long periods of drought. And lastly, this project will provide public enjoyment. Our first research aim in this project was to determine the best seed mix for restoration of coastal sage scrub during low and high precipitation years. To do this, we looked at native species percent cover and species counts in relation to seed mix, year of seeding, and the interaction of the two. In order to address our first research aim, we answered the question how percent cover and counts of native species with different physiological traits varied across native seed mix, year, and their interaction. Our second research aim was to determine if herbivory was playing a key role in the difficulties of restoring our native grasslands. To complete this aim, we placed herbivory exclusion fencing in our grassland plots and performed statistical analysis to determine if herbivory is affecting the performance. To answer our second research aim, we asked how herbivory is impacting the grassland restoration blocks and the survival of Stipropulchra and Cicerinium bellum, both native California grasses. Here we have different seed mixes that we use in our experiment. Each of these groups are based on their functional traits of either high water use efficiency, low water use efficiency, and control of all seed mixes together, species that did well at another restoration, and local species for which we have no previous data on. In designing this experiment, we used a randomized block design. There were 7 coastal sage scrub replicate blocks. Within each block, there were 3 years of treatment, and within each of those years, we added in a random array of our seed mixes to one of 5 plots. Within each of our plots, a designated point for species counts was used for our early season data collection. These points were denoted with ground whiskers. Lastly, we had negative controls for each replicate where we did not do any seed addition. Our first data collection method we used was a point intercept along a transect within each of our treatment plots. Each transect had 28 points. We collected data on which species were hit and whether the species was alive or dead, along with the ground cover. Our second data collection method for the coastal sage scrub experiment involved using a 1 meter by 1 meter quadrat at each of our pre-designated points in our treatment plots. Within each quadrat, species present and the number of species were counted. Similarly to the coastal sage scrub study design, we had 7 randomized grassland blocks. However, these blocks had year and weeding intensity treatments instead of year and seed mix. Within each of these blocks, we placed our fencing in year 2 medium intensity weeding plots. Within our plots, we installed 3-foot tall exclusion fencing. Each of these fences were of a circular design and had a 4-meter diameter. Exclusion fencing was constructed using chicken wire, garden staples, and bamboo stalks to hold the fencing up. Red flagging was used to denote Stipropulchra and Cicerinium bellum that were monitored. In total, 32 Stipropulchra and 9 Cicerinium bellum individuals were monitored out of the 64 and 9 individual plugs, respectively. When we monitored the Stipropulchra and Cicerinium bellum, we collected data on each individual's height, width, whether they were alive, if they were fenced, and if there were any signs of herbivory. These signs of herbivory included squared off, serrated, or discolored edges. Here you can see what the field conditions were like when we collected data on the individual Stipropulchra and Cicerinium bellum. And I would like to now hand over the presentation to Maryam, who will go over our analysis and results for our first research question regarding the best seed mix for coastal sage scrub restoration. Thank you very much.
Hello, my name is Maryam Ibrahim, and I'll be discussing the statistical analyses and results from seasonal data that we collected in CSS plots at the bowl. So in this study, we asked the question of whether water sufficiency functional traits can predict native plant performance during low and high precipitation years. And here we quantified plant performance in two different ways. First is plant density counts, which are quadrat measurements that sample early spring germination. Second, we measured percent cover using the point-intercept method along a 28-point transect to sample growth as observed in the late spring season. But first, we want to establish the extent of interannual variation in precipitation at the bowl and what that means for our study. So our first graph over here depicts rainfall data that we obtained from a local weather station near our study site, specifically in Corona del Mar, and we show that monthly rainfall averages recorded from the first wet season were abnormally low and characteristic of drought conditions, whereas in the following year our study site received more than double the amount of precipitation observed in the previous wet season. So here's where the study of plant functional traits can really offer new approaches for restoring disturbed ecosystems like the bowl. Species that have high water sufficiencies are able to cope with drought because they have certain physiological adaptations to water deficits, such as a deeper rooting depth, delayed germination, low relative growth rate, and conserved metabolic processes. On the other hand, low water sufficient species have higher water requirements and as a result are not well adapted to drought stress. Although these species do have other advantages of being faster germinating, faster growing, and overall accelerated metabolism that enable speedy growth and development in the presence of adequate precipitation. So to that end, we hypothesize that the high water sufficient traits will be favored in the drought year, and we expect to see higher plant density counts and percent cover measurements. Similarly, we expect that low water sufficiency species would have higher performance in the wet year due to increased water availability, presumably triggering that rapid growth across these plant communities within treatment plots. So we performed a two-way mixed model ANOVA with two mixed factors, which are treatment and year. Treatment has 6 levels corresponding to 5 native seed mixes and then no treatment control plots. Year contains 2 levels, including the dry year 2018 and the wet year 2019. We also factored in randomized block design to control for the error that would otherwise be attributed to variation across the landscape. So we carried out these tests twice, once using plant density as a response variable and again using percent cover as the response variable. And so in our first graph over here shows that the all CSS species seed mix, which contains all of the functional traits in the study, had the highest plant density compared to all other treatment groups, which were consistently lower in comparison. This next graph over here, we're looking at the same analysis done using percent cover as a response variable, and right off the bat we see that the highest measurements again were observed in the all CSS species seed mix, but followed closely by the West Loma drought survival and the high water sufficient seed mix. So in summary, we concluded that in 2019 the all CSS species seed mix had significantly higher performance of plant density and percent cover, followed closely by West Loma and the high water sufficiency in terms of percent cover. And so based on these results, our next step was to evaluate species composition and their individual abundances within these treatment plots to find out how individual species interacted with precipitation and which species could be driving this performance. So this graph over here shows you that 6 out of the 21 observed species in the all CSS species mix had higher percent cover in the wet year compared to the dry year. And in this next graph, we're still looking at the all CSS species seed mix, but evaluating percent cover, and we see that Deynandra fasciculata and Artemisia californica both had significantly higher percent cover when precipitation was high. We see a similar result with the West Loma drought survival, where Artemisia californica and Deynandra fasciculata again are higher abundances in the wet year. And lastly, looking at the high water use efficiency seed mix, this time the Deinoundra fasciculata was the only species in that seed mix that performed higher in the wet year compared to all other species in the mix, despite being known for having high water use efficiency traits. And so, in conclusion, we found that species with the highest plant densities were an equal mix of low and high functional traits. And with that, I will hand it off to Peter, who will discuss the implications of these results and possible management recommendations for future restoration of the site. Thank
Okay. Thank you for that introduction, Maryam. So yes, I'm Peter Lamb, and let's get right to it. To reiterate from what Maryam has said, the all CSS species seed mix came out as the only significant seed mix in terms of plant density. And this makes sense since an equal mix of all native seeds provides an opportunity to include as many functional traits as possible. Of particular note are two shrub species, Artemisia californica and Salvia mellifera, that perform well under low and high conditions. Past research indicate these shrubs have traits that support fast germination in high water conditions and allow them to persist in low ones. These findings indicate A. californica and S. mellifera should be major target plants for restoration at the bowl. Another species of interest from the seed mix is Plantago erecta, an annual herb. P. erecta is good at preventing the spread of non-native plants and is a good target plant for restoration. It germinates quickly and is known to compete well with non-native grasses. With significantly higher percent cover in more than one seed mix treatment, A. californica is one of the strongest performing species. Other studies support our findings by labeling A. californica as an opportunistic species with the ability to colonize quickly. Studies have shown A. californica to have a high relative growth rate and a high degree of trait plasticity, allowing for it to capitalize on infrequent rain events for maximum growth and fitness benefits. Another key species with significantly higher percent cover is Deinoundra fasciculata. One possible explanation for this could be the plant's root strategies. Past studies have measured the root responses of various CSS species and classified D. fasciculata as a rapid response type. As a rapid response type, the root elongation rate and the root tip appearance of D. fasciculata responds rapidly after a precipitation event. The rapid combination response would allow D. fasciculata to react to any rain event with quick root proliferation. So the interannual variation in precipitation proved to greatly impact the results of our studies, and analyses indicated that these differences between native seed mixes were only observable in the presence of adequate rainfall. And here's a chart shown earlier of average monthly precipitation in the site with months on the x-axis and rainfall on the y-axis. We can see here that the site received over double the rainfall average in 2019, which resulted in substantial native cover, whereas in 2018 we only observed low values. The adaptations that plants gained from functional traits were not enough to safeguard them from periods of severe environmental stress, so we conclude that varying amounts of precipitation significantly impact overall plant performance beyond what the measured functional traits could provide. We suggest that the functional response of these species may be very context dependent, involving resource availabilities such as water and nutrients, and some of these factors are often cited as being able to predict community level responses to global climate. The relationship between functional traits and plant performance at the bowl is therefore nuanced by changes in the abiotic and biotic environment. Thus, we suggest that these changes interact with the climate to create challenging conditions for plant survival, as seen in 2018. Our study does support our hypothesis as it relates to species with low water use efficiency traits having the greatest performance during the wet year. However, it does not support it as it relates to the low precipitation conditions seen in 2018. The reason being that we could not gain quality data from low or non-existing values in the dry year. To reiterate, we believe the drought conditions of the site in 2018 were so harsh that even plants with well-adapted functional traits found it difficult to survive. And while we focus on water use efficiency as our primary trait, there are surely other functional traits in play. Understanding plant functional traits is a great method for restoration, but it will require a more holistic approach, such as measuring more functional traits and considering the context dependencies. Nevertheless, We have identified the all-species seed mix as the best suited seed mix for restoration at the bowl, and with topography and plant performances that vary by plot, we recommend broadcasting the seed and letting the species sort themselves out as the most efficient method for restoration at the bowl. And what this means for our community partners is that we have assembled an essential plat list based on this mix, with particular emphasis on A. californica, D. fasciculata, S. mellifera, and P. erecta. And researchers looking to continue or replicate this experiment in other areas may benefit from thorough research into germination data and soil sampling, as well as maintaining an inventory of all species of interest, complete with native or non-native classification and a key for naming acronyms. Future studies can expand on this by considering possible reference sites and seeing what would happen over a long period, such as 5 years. I would now like to introduce Justin Morgan, who will be presenting on the herbivore exclusion study.
Thanks, Peter. For the data from the herbivory study, we used an unpaired t-test for the normal data, Mann-Whitney U test for the non-normal data, and we also assumed that all the dead plants were dead as a direct result of herbivory. For the Sisyrinchium bellum, also known as California blue-eyed grass, we saw that the fenced plants were on average 179.25 millimeters taller and 6 1/3 millimeters wider than the unfenced plants. If you take a look at our p-values, you'll see that the difference in height was statistically significant, but that the slight difference in width was not statistically significant. So we conclude that for this species, herbivory did have some impact on plant health as measured by plant size. Looking at the herbivory rate for Sisyrinchium bellum, we saw that about 31% of the fenced plants showed signs of herbivory, compared to 100% of the unfenced ones, which is a statistically significant difference. But when we looked at plant mortality, while the difference of nearly 12 percentage points appears large, the observed difference between the fenced and unfenced plants was not statistically significant. However, we believe this to be a type 2 error, or a false negative, and I will explain our reasoning later in the video. Changing species to Stipopulchra, or purple needlegrass, we see the fenced plants were nearly 255 millimeters taller than the unfenced plants, while also being almost 14 millimeters wider. Again, we see a statistically significant difference between the height of both groups, but this time we also see a significant difference in width. Based on this, we know that herbivory significantly impacted plant health of the species as measured by plant size. For herbivory, once again, we see that all of the plants outside of the fencing showed signs of herbivory, compared to 28.57% of the fenced plants, which is a statistically significant difference. Unlike the previous species, none of the fenced plants died, while we lost over a fifth of the unfenced plants, which is a statistically significant difference. Based on these results, we can clearly see that herbivory impacts the success of Stipopulchra in the context of our restoration efforts. In the notes section of our data sheets, we collected data on which plants were in bud, in flower, or were producing seeds. Obviously, we need plants to be reproducing in order to restore the site and establish a healthy population and seed bank. We found that none of the unfenced plants for either species had reproductive bodies, which was not the case for the fenced plants. This further emphasizes the impact of herbivory on our grassland restoration efforts at the bowl. We did have a disruption to our study due to COVID-19. Because of the situation, weeding efforts at the site came to a halt. Because of this, our replicates ended up being overgrown with Brassica nigra, or black mustard, a common invasive species in our area. In the left picture, you see replicate 1 as it was in February when we collected data, while in the right picture, it's the same plot in April when we went for our second round of data collection. We believe this negatively impacted the health of our monitored plants and may have had some impact on herbivory as well. Because of our replicates being so overgrown, we were unable to locate all of the plants we flagged for our study. While we did take this into account while doing our analyses, the loss of significant numbers of individuals certainly impacted our findings, particularly with regards to Sisyrinchium bellum, which had a small sample size to begin with. This is why we believe that the lack of significant difference between the fenced and unfenced plants for that species is a false negative. Based on our results, we believe that herbivory is one of the reasons that we have such difficulty restoring California native grasslands at the bowl. We are able to demonstrate significant impacts to plant size and reproduction for both species, and that herbivory does impact the survival of Stipropulchra. However, given the high amount of variation between the results of our replicates, we believe that there are other factors at play, likely abiotic in nature. These factors are also likely playing a role in the challenges we've been facing. Given what we have found, we recommend that an immediate change be made to the ongoing study at the Bowl, specifically the construction of herbivore exclusion fences around the Year 3 grassland plots. We also recommend that any future efforts to restore California native grasslands to the Bowl should include efforts to protect the plants from herbivores through whatever techniques such as fencing, plant cages, or rabbit repellent are deemed appropriate for the project in question. On behalf of the entire team, I'd like to thank you for watching our presentation. If you have any questions, please contact our advisor, Dr. Sarah Kimball, at the UCI Center for Environmental Biology. Thank you again.